We are really excited to kick off our speaker series, Nature Matters. As you may know or are now being introduced, Nature Matters is a lively conversation centered around the intersection between nature and culture. We try to bring you really riveting topics that our local community cares about and can learn something new. This is a speaker series hosted by the Lewis and Clark National Historical Park in partnership with the North Coast Watershed Association, the Lewis and Clark National Park Association, and the Fort George Brewery. My name is Kayla Furman. I work at the park, and I am excited to take you along this season. We have a lot of great speakers ahead, but first, I'd like to introduce one of our partners, Graham. Also a salmon.
Hello everyone, my name is Graham Clagg. You also might know me as Sam the Salmon, but I am the Executive Director of the North Coast Watershed Association and very happy to be suited up for you tonight to talk about a less scary subject, which is not Halloween, but Chum Salmon Reintroduction. And very excited to introduce Kelcee Smith tonight, who's the Reintroduction Coordinator for ODF&W, who I've had the chance to work with on moving chum throughout the watershed as we do this about this time of year. And she's going to tell you all about the role of our partnership, the role of habitat for chum salmon, but also how you can get involved with supporting chum and supporting our efforts as we try and move onward and upstream. So thank you guys so much for being here tonight, and have a great time.
It's really hard to follow a salmon. I'm going to do my best. My name is Kelcee Smith. As Graham said, I'm the Chum Salmon Reintroduction Coordinator for the program to restore Oregon's chum salmon, and that's a program that is run by the Oregon Department of Fish and Wildlife. I'm really excited to be here. I think this is one of the coolest venues that I've ever given a talk in, so here we go. Before we get started, I do want to thank my team. I don't do this project by myself, so I want to thank Scott Kirby, Brian Liebersite, Kevin Hall, Eric Cerny, Dr. Chris Hummel, and Derek Wiley. All of these people have worked really hard on this chum reintroduction project over the years, and I couldn't do any of this without them. So I'd like to just kind of point out my title here. It's a pretty provocative question, right? Is 2 years of reintroduction effort enough to restore Oregon's chum salmon? Of course it's not. I think a lot of us in this room might be out of a job if it could be taken care of in 2 years. So hopefully today, after my talk, you will understand why it's going to take longer than 2 years. But first, a little bit of background. Chum salmon are the most widely distributed salmon in the Pacific Ocean. They range from Japan all the way through California. But today, of course, I'm going to be talking about the chum in the lower Columbia River. Chum salmon exhibit a fall run timing cycle, and so what that means is in the fall, they come past Astoria up the Columbia River to spawn. Their eggs incubate in their natal streams over the winter. And then in the spring, the fry out-migrate to the estuary, and eventually they'll go into the ocean. Now, unlike other salmon that we have, chum salmon fry don't spend a lot of time in their natal streams. They move out into the estuary really quickly. The ones that out-migrate from Big Creek can get to the estuary in 2 to 3 days, so they're gone. At this point, they spend the rest of their time in the ocean as adults for about 3 to 5 years until they come back in the fall and start the whole process over again. Now, historically, in the lower Columbia River, chum salmon were super common. In 1928, it was estimated that over 1 million adults returned that year, and that represents anywhere from 7 to 10% of the total salmon return in that year. And you can imagine that before Bonneville Dam, they used to get really far up the Columbia River. And they were really popular for commercial fisheries, as you can see here in this old advertisement for canned salmon. But they were popular too for recreational fishers. This is a photo from the Oregon Historical Society. But this harvest was also one of the reasons why they declined in the river. So this is a graph of the chum salmon harvest in the lower Columbia River. And you may not be able to see the numbers exactly, but the harvest was really from the the early 1900s until about the 1950s. And the whole reason commercial fishermen switched to fishing for chum salmon is because the Chinook salmon population collapsed right before 1900. If we zoom in a little bit here, starting in the 1950s and 60s, we have some more data that shows us about the harvest of chum in the lower Columbia. This data in particular is from the 1970s and 1980s. And you can see how it declines even further. In this first graph, we were talking about thousands of pounds of fish. In the second graph, we're talking about individual chum salmon that were caught. So you can see how the numbers have declined over time. Also, I'd like to point out this million mark, where that was the year that 1 million chum salmon potentially returned to the river. And by the 1990s, we're talking about hardly any returning. Another reason for chum salmon decline in the lower Columbia River is habitat loss. This is a pre-developmental land use map within the historic distribution of chum salmon. And as you can see, it's mostly forest. There's some grassland and other shrub type areas there too. This is what it looks like today. It's been heavily modified. There are still some forests, some late successional forests there, but there is a lot of agriculture. That big black area in the middle is the city of Portland. And there's other residential areas around as well. So all of this change in land use over time increases pollution, it decreases water quality, it means that more fine sediment is being pushed into these systems, and if there are fewer trees around our streams, it means that the temperature in those streams are increasing. That increases a whole bunch of other things, including parasites and bacteria and other stuff that can affect the whole watershed, including chum. Another piece of this habitat loss is the construction of Bonneville Dam. The dam really altered the hydrology in the river, or the water flow. So this is a hydrograph of the water discharge throughout the year. And so you can see the months along the bottom, and then you can see the amount of water that is leaving the river in cubic meters per second. Now, cubic meters per second doesn't really mean a lot to me. I don't know about you. So I converted it to millions of gallons per minute, but that still doesn't mean that much to me because it's such a big number. So I thought I'd pull from something a little closer to home. If we filled Lumen Field all the way up with water, the peak discharge in the Columbia River historically will be not 1, not 2, but 2.5 lumen fields of water every minute. So I failed to mention that this black line here is historically what the flow used to be like before the dam went in. The gray line is the flow that we have now. So we go from 2.5 lumen fields down to 1 lumen field basically today. I hope that helps. The other thing I want to point out just about water flow really quickly is that there's a lot less variation now than there used to be. It's a lot more controlled, as it should be, because there's a dam there. But all of this affects migration, not only of fish coming up the river, but also fish like chum moving down the river. Those little fry can't really swim against too much, and so if there's more water pushing them down, it means they might get to the estuary quicker, for instance. When you have less water in the river, there's less habitat for fish, and there's less quality habitat for fish as well. So what happens when we have all of these threats combined is that we get ourselves caught in this negative feedback cycle. So when we have overharvest or poor or lost spawning habitat and other impacts like predation or climate change or disease or ocean conditions, when we put all of that together, it means that there are fewer chum salmon returning to the river. And when there's fewer adults, it means there's less spawning. When there's less spawning, it means there's fewer eggs. When there's fewer eggs, it means there's fewer fry. When there's fewer fry, it means there are fewer adults, and then we just get caught over and over in this circle. So because of this, 90% of historical chum salmon populations in Oregon are gone. Because of that, chum salmon in the lower Columbia River were listed as threatened on the Endangered Species Act in 1999. Today, we have 8 recovery populations, which are these colored areas here, blues and the greens and the gray areas. There are also Washington populations that have been designated and listed as threatened under the Endangered Species Act, but I'm not going to talk about those today. But because chum occupy the lower Columbia River, between 2 states, they're a federally listed species. It means that there's a huge collaborative effort for their recovery between a lot of different agencies. So the recovery approach that we have at ODFW is a 4-pronged approach. It begins with habitat restoration. It also includes maintaining a conservation broodstock, monitoring any recovery from our reintroduction efforts, and identifying and addressing any limiting factors. Now, today, I'm going to touch a little bit on all 4 of these, but I'm mainly going to be talking about how we maintain our conservation broodstock and how we monitor recovery. So just really briefly on the habitat restoration piece, habitat restoration goes, we don't do a ton of it at ODFW, at least not in our particular project. Instead, we partner with local groups like Return of the Reds to work on local projects that can really be focused on key areas, either to maintain habitat that is already looking pretty good, to add or restore new habitat, to increase the amount available for chum salmon, or to look for projects in the future that can really expand us even further. So I'm going to move on to our conservation broodstock, which is the bulk of our work. A conservation broodstock is basically just hatchery-raised fish that are used for conservation and not harvest. So when you think of a hatchery, you might think of us raising fish to then put out into the population to eventually be harvested. That's not what we do. All of our fish are raised and then released for conservation purposes. So we established our broodstock between 2010 and 2014. And I'm going to zoom in a little bit so that you can see the area that we're in right now. We decided to start our conservation hatchery in Big Creek. And so to do this, how do you start a hatchery from nothing, right? To do this, we took eyed eggs from Washington, their chum population over there, and transferred them to Big Creek. We raised these fish here and released them. Our goal was to release 100,000 fry over 4 to 5 years to really establish our broodstock, with the goal being to eventually transition away from using those Washington eggs and only spawn the chum that are returning to Big Creek. So that's what we did. We keep our chum fry in kind of these ponds here. We raise them to about this size. See how little they are? They're cute. And then we release them via a truck. Our goal was to eventually get to releasing 300,000 fry every year and use any extra adults that were returning as part of our reintroduction efforts, which I'll talk about later. That was the plan. So as you can see, things started out pretty good. In 2013, we were still finishing establishing that broodstock. This is the number of adult chum salmon returning to the hatchery. The green bars represent the number of females, and the orange bars represent the number of males. And so you can see that we really started to get great returns in 2014 and 2015. And during that time, we were able to do some experimental outplanting, where we were able to take some fish and test some different reintroduction ideas that we had. But then, 2016 and 2017, we had really low numbers of fish returning to the hatchery. And this was strange to us because in 2016, you might remember, it was the biggest chum salmon return to the lower Columbia River in 50 years. And none of them came to Big Creek. Unfortunately, this trend also continued in 2018 and 2019. So during this period, we could not do any reintroduction efforts because we could barely maintain our conservation broodstock. And in fact, in 2018 and 2019, we had to go back to transferring eggs from Washington just so that we could continue to support our broodstock. Now, let's talk about 2020. Everyone remembers, this was a record-breaking year for us. In 2020, 978 chum salmon returned to the hatchery, and we were just ecstatic about it. That was until last year, when 2,386 chum came back to Big Creek Hatchery. And as you can see here, even in our best years in the 2014-2015 era, last year blew them out of the water. So last year we did a lot of reintroduction because we had a lot of fish stored. So just to put this a little bit more into perspective, this is the number of eggs that we've collected each year for our conservation broodstock. And you can see it's varied over time, but in 2020 and 2021, we had record-breaking egg collections. To maintain our broodstock, we need to collect at least 110,000 eggs. And to start reintroduction efforts, we need to collect at least 220,000 eggs. That's about 44 females and 88 females, respectively. So you can see that we've only really been able to do reintroduction over 2 years, maybe 3 if you count those experimental years. So we did lots of reintroduction. So while we were incorporating all of these fish into the broodstock, collecting all of these eggs, another 658 chum returned in 2020, and another 1,962 chum returned in 2021. And I'll just say it again for the millionth time. This was a record-breaking year, both of these years for our program. And so we were trying to figure out what to do with all these extra fish. This had never happened to us before. So we had a few reintroduction priorities that we wanted to meet. Firstly, we wanted to try to put these fish, move these adults to great spawning habitat so that when they spawn, they would have the best chance of producing offspring. The second thing we wanted to do is put these fish in places where we already monitor the fry or the adults so that we could monitor these reintroductions, right? We didn't want to just put fish out there and say, good luck. We wanted to see what happened afterwards. We also wanted to put enough fish in a particular stream to actually make an impact. It doesn't make a lot of sense to just put a handful of fish in a stream and just hope they meet each other again. We really wanted to make sure that they were able to find each other. And then lastly, we just had to consider some logistics of moving fish around all of these different areas. So here are our recovery populations again. And in 2020, because we were kind of taken off guard, we decided to stay close with our reintroduction efforts. So if I zoom in on the Big Creek recovery population here, you can see Big Creek, and this is where the hatchery is located. That's the star. And so we moved adults from Big Creek Hatchery just right down the road to Bear and Little Bear Creeks. And in 2020, we put 641 fish total in that system, 400 females and 241 males. And it met a lot of our requirements. Bear Creek has pretty great spawning habitat. We already monitor it. I'll talk about that in a minute. And I mean, I think 641 fish is certainly enough for this system, so that was good. It's also close to the hatchery, which means that the fish didn't spend a lot of time being transported in the truck. It was really safe for us, and they were released lively and well. Another bonus about Bear Creek in 2020 is that we had already observed wild chum already spawning there before we did any reintroduction. So we knew it was a nice, good, chummy place. In 2021, we followed the same logic. Again, we had 1,900 fish come back, so we have a lot to deal with in 2021. So we put about the same number in Bear Creek again to kind of build on our efforts from 2020 for all the same reasons. But we also took fish over to Nat Creek. We took 100 fish over there, 54 females and 49 males. And again, it met most of our requirements as far as good spawning habitat. We monitor it with spawning ground surveys, enough fish, and really close to the hatchery. But this is only 700 fish. We've got 1,200 more to go. So we had to go to nearby Clatskanie River recovery population. This star represents the town of Clatskanie. And we first decided to put fish in Stewart Creek. This is a smaller creek, but we've had some success reintroducing chum in this area before, so we wanted to try it again. So we put 50 fish there. Stewart Creek has pretty good spawning habitat. We don't do a lot of monitoring in Stewart Creek, but we felt like it was a good place to put them. And none of these places that I'm going to show you are really that close to the hatchery, but we had to put these fish somewhere. The second creek that we put fish in was Conyers Creek. We put 168 individuals there. And then we put the bulk of our fish in the Clatskanie River, which is a large system with a lot of habitat availability. We put over 1,000 fish in the Clatskanie during this time. So you can see we really tried to spread these fish out into systems where we thought that they could run into each other, where the habitat looked pretty good. I kind of want to shift gears and talk about maybe what we expected to come back to the hatchery in these years, because they were so big and unexpected. What were we actually looking for in that year, in those years? So I'm going to walk you through a couple of tables to show you how I calculated this out. So for the 2020 return, that is made up of fish that we released, hatchery fish that we released from 2015, 2016, and 2017. So you can see the numbers of fry that we released in those years. Now, what's interesting is that chum have a marine survival rate of 0.2%. So that means if we release 192,000 fry, that we can expect 384 to come back as adults. We also know that not all chum return at the same time. Some return as age 3, some return as age 4, and some return as age 5. Most of our chum return when they're age 3 or 4, so you can see those percentages represented here. So in total, after considering all of these things, our expected return just from our hatchery releases for 2020 was 135 fish. And what we actually got that year was 138 fish. So we were pretty on the money there. If we expand this out and look at the whole return, we estimate that about 20% were from the hatchery and 80% were wild chum in 2020. So we can do the same calculation for 2021. When we run all these numbers out, you can see that we expected from the hatchery releases 264 fish to return, and we actually only saw 207. Now this could mean a couple things. It could mean that fewer survived than what we actually predicted. That's possible. I think it's more likely that we missed some of these fish in our calculation or while we were collecting them on the landscape. When we look at all of this together though, it's likely that 40% of the chum returning in 2021 were hatchery origin and 60%. So these are the calculations for, I'm predicting that we'll get just about 280 fish back. We're just right here at the beginning of our season. We had 1 or 2 chum enter the hatchery today, so they're just starting to trickle in. So we'll see. Next, I want to move on to how we monitor these recovery efforts. And we basically do it 3 ways, and we focus again a lot in these coastal recovery populations. The first way that we monitor recovery is through our spawning ground surveys. This occurs in the fall and winter, and this is literally our biologists going out into the field and walking streams. They're looking for reds, they're looking for carcasses, chum that have spawned already. They're taking a whole bunch of data. It's very intensive. But we do have a select set of sites that are random and fixed across these populations. We also measure fry production in the springtime with screw traps. And right now we're operating 3 traps. This is also really intensive. We have to check the traps every day. We're not only counting chum in this case, we're counting every fish that we catch in the trap. So it's a really, actually a cool way where we record all of the different fish or species in these systems at the same time. We have operated traps in Scappoose and in Youngs Bay before, but generally we can only handle 3 at a time. And then lastly, we can detect chum presence with eDNA or environmental DNA, and we can do that all year by collecting a water sample. It takes about 15 or 20 minutes, so it's really simple, and it can tell us if chum were present within that stream within the last 10 days, up to 1 kilometer upstream from where you sampled. So it's a really easy way to understand if chum were in the system without actually even having to see a chum. So that's super cool. So you can see with all of these different methods that we really monitor these 4 populations pretty thoroughly throughout the year. I want to go a little bit deeper though on how we measure the fry production in our traps. What happened after we outplanted all of those fish or reintroduced all of those fish last year. So these are the outlines of the Big Creek recovery population and the Clatskanie River recovery population. And I'm going to talk about the fry that came out in 2021 and 2022. But before I do that, I just want to mention that the fry that we released from the hatchery came right out of Big Creek. In 2021, we released over 350,000, and this spring we released over 400,000. Again, to give you some context of how many fry we typically release, if you'll remember back when we were starting the brood stock, we were only releasing 100,000. These were our first returns in 2014. We had that really bad period in 2016. We don't like to talk about it. And then in 2018 and 2019, we had those Washington transfers. And then last year was our record. So just a little bit more context about how record-breaking last year was across the board. So moving back to the map, we operate 1 screw trap at Bear and Little Bear Creek. We've been operating a trap there since 2017. And what's interesting about 2017 is that, again, we didn't do any reintroduction in that stream in that year. So this fry estimate that you see on the screen of 61,000 fry is all from wild chum. That's without us doing anything. So in 2018, 2019, and 2020, we didn't see any fry from Little Bear or Bear Creeks. But in 2021, we estimated over 40,000 left the system. And in 2022, over 31,000 left the system. And that was again after reintroducing anywhere from 350 to 400 females. We also operated a screw trap in Conyers Creek. We've been operating a trap there on and off since 2012, but we've never captured a chum fry. So last year in 2022, we estimated 3,800 fry came out of this small system, and that was the first time we'd ever measured that many chum. We also operate a trap in the Clatskanie River, and this is our longest running trap. We've been running it every year since 2012, but over that decade, we've only ever caught a handful of chum fry. Again, until last year, we estimated over 48,000 chum fry left the system. And because we have these multiple estimates for Bear Creek, I want to talk a little bit more about that and what that kind of means for our reintroduction efforts. So I'm going to bring you back to some tables. If we look at the 2020 brood year, we reintroduced 400 females into Bear and Little Bear Creek. We estimate that they would have laid over 1 million eggs. If every female that we put in that system spawned, it would have been over 1 million eggs. But we only estimated 40,000 fry. So really, the actual survival from egg to fry is less than 4%. After you account for marine survival, that means 80 fish would come back after we reintroduce 400. It's a pretty similar story for 2021. Almost the same number of females, the same number of eggs that we would expect to be in that system. But again, our actual fry production was a lot lower than maybe you might expect. So our survival is still hovering around that 3.5 to 3.9%. And after putting 350 females in there, we would only be getting 63 back. So I started thinking, like, okay, if we do this again this year and this fall in 2022 and take an average amount of females that we're reintroducing and their survival and all of that together, okay, we do that again this year. Well, what about next year? Will we do the same thing in 2024? Wait, how long are we doing this? If these 2020 fish are coming back again in 2023 and the ones in 2021 are actually returning in 2021, how long are we gonna have to be putting 375 fish into Bear Creek? So I did a little bit more math. And my main question was, how long does it take to recover Bear Creek? And so in this hypothetical example where we have 375 females that we reintroduce every year with these survival rates, these average fecundity, and the age structure, if everything's kind of normal, and let's pretend that we want to get a population of 300 individuals reestablished in this system, how long would it take? Well, if we just do what we're doing now, it basically doesn't happen. You can see it flatlines pretty quickly. Okay, so what if we slowly increase the number of females that we reintroduce into Bear Creek over time? Let's say we start with 375, but what if we get up to 700 females? Then what happens? Not much. Okay, Kelcee, what if we start tomorrow putting 700 females into Bear Creek? Then what happens? Not that much. What if we change that egg to fry survival rate? Let's double it. We'll make sure that we're just putting 375 females in the system, but maybe their survival is a lot better. I wonder if that will help. We're still not there. We're still at 150 fish. Let's see, maxing out in maybe 2026. If we increase the egg survival rate even more, we're getting closer. And I'll just move on to the next one. If we want to continue reintroducing 375 females into Bear Creek, we would need at least a 14% egg to fry survival rate for us to make any difference in this system. That's with everything else remaining the same, which we won't, no it won't. But assuming these things, we can't just keep adding more fish to these systems and expect it to work. And so I want to point out that with this, estimate, that we would reach this recovery goal in about 2027, maybe 2028. I also want to point out that this would take some consistency, right? A couple of years, 3, 4, 5 years of consistent work in order to see any kind of results. Because what we do today, we don't know the result of until the chum actually come back. It's really hard for us to know if we're making a difference right now. We have to wait for those results. So what have we learned so far? Firstly, maintaining a conservation broodstock is essential. We can't do any reintroduction without that first. We also know that hatchery survival is really high. Last year we collected 424,000 fry. We've also learned that wild survival is pretty low. We expected over 900,000 eggs to be put in Bear and Little Bear Creek, for example, but that only resulted in 35,000 fry. So getting back to this negative feedback loop that I talked about in the beginning, how are we going to break that? To me, there are kind of two ways to look at this. There are probably more, but the first one, which we kind of tried, is to increase the adult abundance. If there are enough adults in these systems, habitat quality may not matter. So let's say we have a situation where we have no chum harvest, we have kind of poor habitat, but there are no other impacts. There could be a large return to the system like there was in 2021. With so many fish in the system, they might be able to clean up some gravel. There would definitely be a lot of spawning, so maybe there would be high survival. That could lead to a more resilient population where we could break this cycle. But we know from what I just showed you that adult returns alone won't work. And it would have to be a really big jolt to the system in order to make a difference. We also know that we can't always depend on a lot of chum to return to the system. If you remember 2016 and 2017, it was really low. There was no way for us to test this idea in those years. So the other way is to increase habitat quality and quantity. And so in this case, if enough quality habitat exists, then short-term impacts may not matter. So if we have no harvest and really great spawning habitat, but let's say we have a year where there's a disease outbreak, for example. We might have fewer adults spawning in the short term, but because there are multiple age classes returning over time, and that really great habitat, those eggs would better survive to fry. And so again, we might end up with a more resilient population and break that cycle. But we do know that habitat restoration is expensive, it's time consuming, and depending on the system, it's not necessarily permanent. These systems are very dynamic. But it is really important to do because it improves the ecosystem as a whole and it provides a longer lasting impact than any other thing that we can do. We also know though that these short-term impacts aren't as short-term as they used to be, or they're not always short-term. So this leads me to our fourth approach that we take for our recovery, which is studying limiting factors. And I'm going to touch briefly on this because I know I'm running out of time. My predecessor did a lot of research on the parasite here in the Pacific Northwest called C. shasta. This is a naturally occurring parasite within our waterways. It's totally normal. It's supposed to be here. There are two types that affect all salmon. The first type affects Chinook, and the second type affects chum and coho. And so what she found by collecting a whole bunch of water samples throughout this region is that C. shasta basically exists a little bit of everywhere. And the type that affects chum can be kind of anywhere at any time. These are her results from 2019, and so in some cases you can see how right here is a green dot where Big Creek Hatchery is. Oh, you can't see. Where Big Creek Hatchery is, there is a green dot, so we couldn't find, there was no parasite there, but right out into the river, there's C. shasta everywhere. So in this year in particular. So if fry are leaving the hatchery and then immediately running into this parasite, it's not so good for them. She found that even small amounts of this parasite are lethal to chum fry. She also found that the higher the water temperature, the more of the parasite there is in the system. And so the main take home from her research is that if C. shasta is present, then chum will be absent. If C. shasta is absent, then chum will be there. What's concerning though about the temperature piece of this is that we know that climate change is affecting a lot of these things. In particular, we know that temperature is only increasing over time, which of course can affect our fry. It can mean that they emerge 2 to 5 weeks earlier, which could also affect their growth, their survival, and any interaction that they have with this parasite. We also know that winter precipitation is increasing and predicted to increase, and we've already seen some anecdotal evidence of lots of water and flooding in the wintertime disrupting chum reds, particularly in 2020 and 2021. And lastly, I'll just say that we haven't even talked about the ocean, which is where where chums spend most of their life. This is the NOAA stoplight chart. Every year they take a suite of climate and atmospheric conditions, physical conditions, biological conditions to create a yearly average of what the ocean is like out there. So the red squares are poor, the yellow squares are medium or moderate, and the green squares are good. And so you can see that in 2014 to 2018, we really had some bad ocean years back to back to back. And that correlates with our poor returns as well in Big Creek. Last year was really awesome though, really good ocean year. So is 2 years of reintroduction effort enough to restore Oregon's chum salmon? Of course it's not. But that's because the spawning habitat quality is pretty low in our area. There's not enough fish that we can reintroduce, at least in 2 years, to make an impact. And there's a lot of other factors that can inhibit our success. But what could work to restore Oregon's chum salmon? To me, we really need to prioritize habitat restoration and reintroduction together, as well as maximizing survival at all stages. If eggs can't make it to fry, then the eggs may as well not exist, right? If fry can't make it to smolts, then the fry may not exist. The third thing that we have to do is monitor. Like I mentioned, anything we do today, it takes 3 to 5 years to see the results of those efforts. And lastly, I'll just point out that this whole thing, it takes a lot of patience. Chum salmon in the lower Columbia River did not decline overnight, and they won't be recovered overnight either. So we have to work hard together in our community over a long period to do this. So I'll just finish here by saying thank you to a lot of people, my crew at ODFW, some of you guys are in the audience. Thank you for all your work. The folks at the Big Creek Hatchery, they do a lot of work for us. Some of our spawning ground survey friends across the river at WDFW who have really helped a lot. We had a lot of volunteers come out last year to help us move these fish all around the state of Oregon, and some of them are in here, so thank you very much. If you're interested in volunteering with us, definitely come talk to me after this. We have spots open. And then lastly, I'd like to thank our funding sources, the Pacific Coastal Salmon Recovery Fund and the Mitchell Act funds. So with that, I'll take any questions.
So if you'll raise your hand, I will pass the mic to you.
Thank you, Kayla. Thank you for such an interesting talk and for all the work you're doing for chum recovery. I have a bunch of questions, but I'll just start with, you spoke of the hatchery versus wild returns. Where are the wild fish from? Are those all Big Creek or strays, or is it not?
That's a wonderful question. So when we first established the broodstock, there were a lot of strays that were going to Washington, right? Because those eggs originated from Washington. But really, we don't see a lot of straying anymore. We know that before we started this program, like one of the reasons why we chose Big Creek is because it has going to have some chum that naturally spawn in Big Creek as it is. So there are wild fish there. Sometimes they get a little lost and end up all the way at the hatchery. It's also possible that the hatchery fish that we've released in previous years haven't made it all the way back to the hatchery and have just spawned in Big Creek before they make it to us. And so perhaps they've created wild fish over time. That's definitely something that happens. So it's a mix of things. It kind of depends on the year. As you can see, we have variation in how many hatchery versus wild fish are coming back. So, yeah.
Thank you. I'll go ahead and ask one more question before I pass it along then, because that kind of leads to one of my next ones is with the super abundant returns in some years where you had so many are looking for other systems to put them in, how much chum spawning habitat is available above the hatchery in Big Creek itself? And what about just getting them there?
Yeah, so I'm not exactly sure how much is available beyond the hatchery. It does get pretty steep pretty quickly, which is, again, not something that chum particularly like. Also, you know, we do want to maintain this conservation broodstock, so in some sense we want them to stop at the hatchery. As far as getting them above that into Big Creek, that's not a big priority for us at this time. It could be in the future, but because that system is so well established and it's kind of the home base for our reintroduction efforts throughout the rest of the state, it's working for us right now to kind of stop them where we do.
Hey, thanks for a great talk. It was
Sure.
really interesting. You mentioned the ocean situation at the end, and you had your interesting model, and the one variable you didn't change was the 0.2% of ocean survival. So I'm wondering if you ever did play with that, and how much would that have to change in order to make a difference in returns?
Yeah, you know, that's a really good question, and it's, you know, the example that I went through is fun because you can change all kinds of things and get all kinds of answers, but of course we're limited with our time here. So I didn't tap into that 0.2% marine survival rate. That is a well-established average in the literature. So it's based on a bunch of different studies over time, and that's kind of the number that we use for chum. I have not run the numbers to see what would happen if that changed, but it is an average, so we know that it does change, and that could be some of the reason why we see, like you said, you know, really big return years and really bad ones. It's probably that marine survival rate. They spend so much time out there, right? Like it's got to be.
Any other questions?
Can you define what a screw trap is for us?
Yeah, sure. I'm sorry. I should have had some pictures. It's basically two pontoons. So it's about the size of like a small VW Bug car, maybe a little bit smaller. It's got two pontoons, and then it has a giant cone that sits in between those pontoons. And that cone rotates as the water moves through the cone. That rotation funnels fish to the back of the trap where they get trapped in a live well. And so they stay there and hang out in the live well until we come and look. And after we collect the fish there, you know, we measure them, we count them. Sometimes we take a genetic sample. So that's how we process them. That's a good question.
Thank you.
So, of the 96 to 86% of salmon that don't make it from egg to fry, do you know what percent are lost to predation and what part are lost to like water conditions and what part just never hatch? Because it seems to me that some of those factors would be much easier to improve than others, because I don't think there's much you can do to increase the amount of eggs that aren't like blighted and just never hatch.
Right. No, that's a really good point. And I mean, when you say it like that, it sounds like no chum fry ever come out of these systems. I do want to point out that we have had higher freshwater survival or natural survival like that in the past. So it's not always, 3.7, but yeah, as far as which factors impact that survival the most, that sounds like a really good study for a graduate student to do that I would be in support of, because I don't know the answer to that specifically. That sounds like a really great question that we might be able to answer in the lab, for example, to see how many eggs actually emerge and hatch to fry under different environmental conditions. So I don't know the specific answers to those, but it sounds like we should figure that out.
Great question.
Yeah.
Thank you so much, Kelsey. Great job. And thanks for plugging Return to the Reds, which is our campaign for Chum Salmon. So if you're interested in getting involved, please sign up here on your way out. But I also just wanted to ask, in terms of potential solutions, where do you see habitat restoration in combination with, let's say, on-site egg incubation maybe to deal with some of these issues? Or kind of what are your thoughts on what that might look like?
Yeah, so I mean, both solutions take a long time. Habitat restoration can take a while, right? Us figuring out where to put these fish and timing these big returns to put them in the right places at the right time, that can be sort of like, we may not know, right? Like, we don't know how many fish are going to return this year. So I don't know exactly where we should be putting them, right? Is this habitat restoration over here far long enough for this year, or should we wait till next year? That kind of thing. So there's some logistics to work out there. But in general, I do think that habitat restoration plays a really big role and maybe should even come first. It's possible that we're able to put enough fish into a smaller stream that has, okay habitat, and that might work out for us over time eventually as it gets, restored, from the list that you have, for example. But I think it might be better for habitat to be restored kind of almost first, or at least get a jump on before the reintroduction part of it. So it definitely goes hand in hand, though.
Great. Well, if you guys want to see where those areas are, check out returnthoreds.com.
Any other questions?
Hey, thanks for presenting and coming out. I was wondering, when there were those really big returns in 2020 and 2021, and we moved fish around, I got to move some of those fish in 2020 into Bear Creek. And I'm thinking about your tables, and I'm like, well, if you just let those fish hang out and spawn in Big Creek, and those numbers went up, and like everything else staying the same, like if you would just let them stay in one system over time, like would those big numbers maybe restore that system and trickle out to others?
Potentially.
Yeah. Would you ever let them stay? Why did you decide to move them out?
So there are a couple of things that we're concerned about here. The first thing is that we don't want to overwhelm any one system, right? So just because, 2,000 fish come back to Big Creek doesn't necessarily mean that it can handle that many fish or that many reds. It doesn't necessarily mean that survival is going to be higher, right? So we could leave the fish there, but it may not result in a positive. The other thing we have to think about is that these fish are highly mobile. So if we take chum that have come all the way back to the hatchery and truck them down to the beginning of Big Creek, they're going to be right back at the hatchery in about 24 hours. So that's just a lot of moving and stress on the fish over time, over and over and over and over, to keep them in Big Creek. The other thing is that we do eventually want to, or we, one of our goals is to move these fish across the landscape and to spread them out like they used to be. And we can only, see if that will work if we try it, right? So that's another reason to think about getting chum out of Big Creek and spreading them out a little bit more. Did that answer all your questions?
Yeah, I guess it's just once they're at the hatchery, you have to do something with them.
You do. Yeah, you do have to do something because they can't just sit at the hatchery, right? So we either have to spawn them or move them.
Could you talk a little bit more about straying with the salmon? I know it's pretty normal for them to come back to the same stream, but you mentioned strays, so I just kind of wondered what that meant.
Yeah, so ideally we'd like to have the chum salmon that we spawn at the hatchery and release from Big Creek. We ideally would like to have those exact fish return to Big Creek, right? But that doesn't always happen. Sometimes they get confused, sometimes they get lost, sometimes they take a wrong turn, right? So sometimes the hatchery fish that we release from Big Creek end up in other areas, and that means that sometimes other fish from other areas end up in our system. I think the weirdest chum we ever got was one from Alaska. So that guy was really lost. This doesn't happen very often, at least not as much as it used to. Now that our bird stock is pretty well established in Big Creek, we don't see a lot of straying like we used to. And we honestly don't get that many chum straying to Big Creek from other areas. So like that Alaska fish was a real anomaly. That's only happened once. So we do keep track of that, but it's not as big of an issue at this point for us as it used to be.
How do you deal with Alaska fish?
Oh, really good question. So we mark chum salmon by putting a mark on their otolith, on their ear bone, and that's done by raising and lowering the water temperature when they're at the egg stage, and so it creates this unique pattern on their otolith. And every hatchery does this marking system on their fish otoliths. And what's really fascinating about it is that each hatchery develops a unique mark, not only for chum, but for every species that they process, right? So if you give me an otolith and I don't know anything about that fish and it's marked, I can tell you what hatchery it came from, what state it came from, what year it was marked, and what species it is. So we got the otolith out of that particular fish, we looked at it under the microscope, and we knew it was an Alaskan fish.
Hey, Kelcee. I just was wondering if you see any competition between chum and other species of salmon in terms of like red development on those streams, or because chum are lazier than coho and chinook if they're kind of safe in that respect.
That is another good question that could go back to what you were talking about earlier, right? A lot of times what happens is when you have a bunch of different salmon in one stream, they're all trying to spawn as they create reds, right on top of each other. And so there is a lot of competition for space. Again, we haven't measured this exactly, but this would be another great study, is to see how much red imposition we're having in these areas, to see if that's affecting it. You know, if a chum makes a red and spawns, and then a coho comes up right after it and messes everything up, well then that could decrease the survival, right? So that's definitely a possibility in these streams. Maybe another thing that's happening even more frequently, which I kind of mentioned, are these big pulses of rain. And you guys remember we had snow in April last year. All of that water flowed off into these streams, very rapidly. And that can scour a bunch of, can scour the reds and disturb the eggs as well. So that's another possibility.
Thank you, Kelcee.